Hello. So, if you're watching this, you've likely been following our series, The Nature Files, where we solve some of nature's biggest mysteries by tapping into the world of environmental DNA. My name is Katrina Pine, and I'm the producer behind the series. We've now wrapped our last case file, but we have one more treat for everyone. I'm here with Matt LeMay, Colleen Kellogg, and Sue Velasquez, three of our research scientists here at the Hackeye Institute, who are our in-house experts on eDNA. This series could not have come together without these people. So Sue, you might recognize from episode 4, the case of the spawning sockeye. And Matt and Colleen here have been behind the scenes fact-checking all of our scripts. And today I get to grill these pros about eDNA. No, I'm just kidding. But I do get to ask you some questions that perhaps didn't make it into the series. So get ready. So, Matt, can you tell us a bit
Right.
about how you come to be an eDNA expert and the work you do with eDNA here at the Hackeye Institute?
Yeah, so I've always just been really, really excited about marine biodiversity, and eDNA provides a really great tool to study all the unknown critters that live in the ocean. And so over the last like 5 or 10 years, I've really just been applying that to a whole bunch of different ecological questions that we can ask with this tool.
So cool. Thanks, Matt. And we're also joined here by Colleen Kellogg in Vancouver. Colleen, tell us a bit about your eDNA work.
Thanks, Kat. I'm a biological oceanographer at Hackeye, and I use molecular tools and eDNA to explore the biodiversity of life in the ocean. And especially the smallest organisms in the sea, the plankton, which make up the base of the marine food web, thinking about who they are and how they're affected by changing ocean conditions.
Oh, okay. And last but not least, Sue. Sue, can you tell us a bit about your involvement with eDNA here at Hackeye?
So my main role is managing and coordinating this integrated coastal observatory environmental DNA work, which involves partners up the coast collecting monthly aquatic samples and looking at fish biodiversity long term.
Awesome. So thank you so much, you guys. All right. Without further ado, I'm going to direct our first question over to Matt. So in this series, we were focusing on solving mysteries in the coastal environment. So a lot of the sampling that we see is all, with water. But can you sample for eDNA in sediment or in the air? And if so, how would that work?
Yeah, that's a great question. So there's DNA is everywhere, and certainly most of the work has been in oceans and lakes and rivers. But recently, a lot of work has been done in aerial or airborne eDNA, and so in that case, we use basically a big vacuum to suck all the DNA out of the air through a filter, and using that, we can figure out what's living in a particular area. And so that's been applied recently to studies of bats. So trying to figure out what bats are flying around at night, you can vacuum out the DNA from the air to solve that problem. There's also been some really neat recent work looking at eDNA on spider webs. So you can take all the eDNA from spider webs to figure out what the spiders have been eating. And also recently looking at rainwater flowing over trees to figure out all the insects that live in the trees. And so really there's DNA everywhere, and with the right tools you can study it and figure out what lives there.
Thanks, Matt. So for the episode, The Case of the Spawning Sockeye, we looked at how a community was gathering eDNA data to try to restore salmon populations in the Fulmore Lake region. And this question is for Sue. So how else can eDNA be used to affect policy, management, or conservation?
I'd say, yeah, if the presence of certain species of interest in an area can maybe help strengthen the case for establishing new marine protected areas or indigenous protected and conserved areas. I would say a lot of communities are concerned with invasive species. So if we're able to detect those, that might raise some questions about conservation. As well, the aim of our project is trying to monitor fish biodiversity long term. So if we're looking at those trends and sort of changes in fish biodiversity, a decline in diversity could maybe indicate a decline in ecosystem health and maybe create the need for better conservation measures in those areas.
So now we have a question that I'm going to direct over to Colleen. So one thing that wasn't really discussed in the series is the cost of doing eDNA analysis. We talk a lot about these like fancy machines that we're using in some of the episodes, but how costly is it to extract and amplify and sequence eDNA? And is this becoming more accessible anytime soon?
Yeah, so those instruments are definitely pretty expensive, over hundreds of thousands of dollars. So the upfront cost can be pretty expensive to get to sequencing in your own lab. But the cost to sequence DNA overall has really dropped tremendously since we started using these sort of approaches over the past like several decades. Um, so it might cost, I don't know, maybe somewhere between $10 to $15 to extract the DNA from a sample, depending on kind of what method you use. And then another several dollars to make copies of like that gene of interest to tell you about the diversity of the particular group of organisms you're interested in. And then you get to the sequencing costs, and that might cost somewhere, I don't know, like around $3,000 or so to sequence samples, but you can mix a whole bunch of samples together. So like you can mix over 300 samples together to run them. So if you think about that, $3,000, 300 samples, it might be around $10 or so a sample to sequence that sample. But then if you think about the fact that you're getting many gigabases of data from these sequencers, you end up only paying basically a fraction of a cent per base pair. So then it costs, I don't know, in total, maybe somewhere around $40 to $50 a sample.
Wow, so actually more affordable than I would have thought. That's really, for such fancy machines, that's really amazing. So I'm going to direct our next question over to Sue, who has done a lot of the training with our collaborators, training people to do sampling and filtering for eDNA out in the field before sending those samples back to the HackEye Institute for extraction, amplification, and sequencing. So Sue, how hard is it to teach someone to do eDNA sampling, and are these sorts of collaborations something that we could be doing a lot more of?
Yeah, it's actually very easy to train someone. I think from the start of this project, we just developed some methods that we thought would be accessible to everyone, and we've had some great feedback. So it has been very easy to train people with no prior eDNA background in sampling in the field, and they've been able to kind of pick up or learn the methods and execute them very, very quickly. They also don't have to be taxonomic experts, and it's also less time consuming than other survey methods. Yes, one real advantage eDNA has over another study method, like say beach staining, is that there's an opportunity for a lot of citizen science. So a larger number of people can contribute a much wider spatial scope.
Okay, so our next question is for Colleen. So can eDNA really detect like super small things like larvae in the water or even viruses, or could it maybe one day?
Definitely. We're like that, like this is a perfect question for me. um, well, for sure, basically eDNA is detecting any sort of genetic material in the ocean. And actually most of the DNA we get when we filter a water sample is DNA from the microbial life in the ocean, because we're actually capturing those organisms on our filters. And so eDNA really stems from the field of microbial diversity and looking at all these microbial tiny things in the ocean. And so it's perfect. It's a perfect tool for looking at larvae, which are small, bacteria, microbes, other like phytoplankton. And also we can use it to look at viruses in the ocean as well. And we've been doing that for some time. Kind of sometimes what you have to do is change the size of the filter that you're using to collect all that DNA from the ocean. But other than that, you can really target kind of whatever forms of life you want. So,
Colleen, I knew you were the perfect person to answer that question. And so this brings us really to our last question for today, which I'm going to direct over to Matt. And this is all about the future of eDNA. So Matt, I want to know, this is going to age me a little bit, but I want to know, will the Star Trek tricorder, like, handheld, instantaneous eDNA results, will that ever be real? I need to know.
Yeah, that's a really exciting question. I think yes to your question. I mean, we've made really great progress in terms of portable, rapid DNA sequencers that we can use that are almost the tricorder that you're talking about. And I think in the near future, I can imagine something kind of like a COVID test, where you put seawater on and it can tell you if a particular invasive species is present in that area or that sample. And I think the field is going that way. But I think there's also the future is exciting for what we can get out of the data too. So for right now, we can tell if a particular species, say sockeye salmon, lives in the area, but we don't know if it's an adult or a juvenile, because we don't have any life stage information. And I think in the near future, we'll be able to start getting more information about the type of the fish and potentially also population. So where did that fish come from? Is it a Fraser River sockeye or Columbia River or something like that? And so I think both the speed and the amount of data that we'll be able to get is going to rapidly change in the future.
You heard it here first. If the Star Trek tricorder is not a real thing in 5 years, I'm going to be very disappointed now. Well, that brings me to the end of our questions. Thank you so much for doing this Q&A with me today and for your help on this series. Our hope for this little series was that it might inspire people to think about what eDNA might be capable of going forward. So I hope that we've done that, and I'm excited that we're finally able to share all this exciting research that's been going on these last few years. So now I'm just really looking forward to seeing what happens next for eDNA. So thank you all, and signing off.